Okay, I think that what we'll do, we'll just make a start now. First of all, a massive thank you for anyone who has joined me today. I appreciate it because we have a couple of important things to go through. And as the very first thing, what I want to do is just to kind of open the floor for any questions that might be related to anything else than the 45-day count, which is coming up, or the exam that we have scheduled for this week. So feel free to just unmute yourself and shout out if you have questions about anything else but those two things, the exam or the 45-day count, because I will cover both of those in just a moment. And as always, I do realize that we have a rather limited time together today, so that's why I'm focusing on making sure that we make that time count. I heard some messages coming through, so I'm just gonna check those. Will there be a study guide? Yes, I just released the study guide maybe about an hour ago. So that's what we're doing today. So if you haven't had a chance yet, you will find the study guide under the course content within the between the modules for week 6 and week 8. So we're on week 7. So over there, you will find the study guide for exam 2. And as always, or as before, the study guide is full of blanks. So that's really what I'm hoping to do today with you, to go through those blanks, what you might expect to see, what sort of things are going to come up. You'll also find in that space between the week 6 and week 8, lecture exam 2, and that is the exam that I'm asking you to take. That was a very good question. I don't see other questions coming through. If you think of something, just unmute yourself. But while I'm waiting for those, I'm just going to jump back to where we were earlier on, because I really do want to make most of the time that we have together today. And I'll take the attendance in a little bit, but before we take that, there's a couple of things that I do want to do, and I want to use some of those activities as a means of also taking that attendance. So as some of you who have been with the college a little longer know, one of the processes that we do 45-day count as every semester as a way of being in compliance with the federal regulations for reporting. And what the 45-day counts does, it helps us to make sure that the students that we are having in our class are actually recorded there, and any students who are not in our classes, who are not participating in our classes, we do not want to report them as if they were attending. And the 45th day of teaching on this semester falls on this week's Wednesday. So on Wednesday, pretty much every instructor, with the exception of some late start classes and shorter classes, but pretty much most of the instructors on this college will be weeding out through their rosters for every class. And if we haven't seen you around taking part, and taking part at least once a week, there is a very real chance that you might be just dropped from the class. The other way you might be removed from the class, which I prefer, is that the student themselves asks to do so. And I'm not the person who can do it for you. You have to contact the registry and ask to be removed from the class. And if you do that, I believe until the Wednesday, you stand a good chance of not getting a poor grade on your grade record and so on. So you really want to make sure that you're kind of starting to consider that, am I completing this course or am I not going to complete? It is not, though, to say that you can count on all of us doing this. Some of us do it a little bit more actively than others. So if you are not participating, there is a chance that you might still sneak through the process and be recorded as participating on the class. And it really is in your benefit to make that call and get removed. It's not something we can put on all of our instructors. So if the class is not working out for you, this is the time to reach out to the instructor. I always reach out to my students before I drop anyone, and we have a little bit of dialogue about that, but I can't promise that every instructor would do the same. So just something to keep on mind, and especially check those emails, check those Blackboard messages this week if you have not been actively participating, to make sure that if you are getting an email from your instructor, it might be very well about this. So that's all I can think of to say about the 45-day count. Do we have any questions about that? If you're taking part on class, things are going well, you're getting points populating on your grade record, it really requires no action on your behalf. You're doing well if you're seeing grades coming through. If you're not seeing any grades coming through, if you're not contacting your instructor, if you're not having any dialogue, then I do really recommend at least keeping an eye of those messages. I don't hear any questions regarding that. So the next thing that I wanted to speak about is that, what are we doing this week? This week has been dedicated completely for the exam 2. So exam 2 is due on Saturday at the end of the day of this week, and it covers 3 chapters: chapter 3, chapter 4, and chapter 5. And as you'll see, that's the only real thing that I have scheduled for you for this week. We do have this session today, and if we get through everything today, this is the session we have. If we don't get through everything, then I have penciled in time on Wednesday, but I really do think that going through the same study guide twice does not add any benefit for you. You might instead on Wednesday just sit down and get that test done. So I'm leaning towards of not having a class for us on Wednesday, and rather encouraging you to take that time or another time during this week to make sure that you get that exam in. The exam has been scheduled for 2 hours. That's way above the time that you need for it. There's going to be 50 questions. In total, it's worth of 80 points. So aligning very well with what you already know. A similar format. So you should be able to answer to each question. You'll either know the answer or you do not. So you don't need all that time to answer those questions. I'd say that in about 15 seconds, you'll know whether you know an answer to a question or not on average. So it is an open book test. There is not going to be proctoring required. You can use your notes. You do want to be prepared for it, though, because if this is the first time you start looking any of the course material, then the 2 hours might be pushing it there. And that's the way there is time, or the reason why there is a time limit, that this is not the time to start for the very first time figuring out what biology is and what biology studies. However, even though some people find that time intimidating, the timer, I can promise that you'll have more than ample of time to tackle the questions. And in fact, on today's session, it is my goal that we get to go through every single question that might come up in the exam. I won't say that these are the 50, but we'll cover all of the material. And amongst that material, every single question type should be covered as well. So that's the plan for today. And before we jump into the actual exam review or exam study guide, I just want to make sure if anyone has any questions, you can just unmute yourself and speak up, or you can text to the chat. And I will also take a few moments to discuss what the extra credit question will be. So there's no curveball about that as well. You will know what it is. But before we talk about extra credit question, do we have any questions about the exam itself or things like that?
I have a question.
Yes, I'll try to answer.
Okay, so on the exam, would some of these questions be like 3 to 5, the quizzes questions on the exam?
It would be, very much so.
Okay, thank you.
Yeah. Good. That was a good question. So I really do encourage you. I get often asked by students that what should I study and can I get a study guide? You probably have seen all of the questions already. I won't promise that, but you probably have. So it is the exact same format. When you took the quizzes, you have had multiple chances to take those, and you have had a chance to revise the correct answers to those. So there's no reason why you wouldn't get 100 points for this quiz or this exam. That was a very good question. Sounds like you figured out my way of approaching teaching. Any other questions? I don't see other questions coming through. So let's talk quickly about the extra credit question, because it's a great way for me to also get the attendance for this class. So let's remind ourselves. We talked earlier on this course about proteins and how proteins can be studied at various different levels of detail. And there were 3 levels of structure that we highlighted for the study of proteins. First of them was the primary structure, which referred simply that we're studying the amino acid chain. So the sequence of individual amino acids that we have placed one after another based on the coding from the triplets of bases in the DNA or RNA. So protein on this level, considering a protein, what protein is on this level, is nothing but a chain of different amino acids linked one after another. You can think of it a little bit like as a pearl necklace or whatever you wish. So it really is just a clear order of amino acids placed one after another. So that's our primary structure of proteins. Secondary structure of proteins now steps a little further, and we're actually, instead of looking at these individual amino acids, we're looking at the creation made by the chemical formulas that we have created. So we can use this chemical formula as a starting point for visualizing what the protein will look, either as a sheet or a certain kind of a helix, kind of a ribbon, if you wish. Whatever we prefer to do, it will show us that proteins have this kind of a one-dimensional shape, or shape that's very close to being a piece of paper. You can twist off that paper, it can be many different shapes, but at large, on the secondary level, we're simply looking at it as a structure rather than these individual amino acids, but structure that has some amount of three-dimensionality, but I would describe it much more as a two-dimensional structure on the secondary structure level. And the tertiary structure of a protein now steps into this really fully 3D world, if you wish. So what we're seeing here is a good example of what protein can be viewed as on a tertiary level. So here it's taking this globular shape. So rather than just being two-dimensional sheet that might be bent, that might have been pleated, now we're often describing these three-dimensional tubes or structures that we can visualize often on computers, for example, and rotate them on the screens. So now we're moving to the truly 3D view of the proteins. So we moved all the way from amino acids to actually a fully three-dimensional shape-filling or space-filling model of the protein. So I hope that that makes sense. And if you're really kind of confused, where does that come from? Remember that the amino acids provide us understanding of the chemistry, the chemical composition, which we have drawn out here. And we also have drawn out a helical shape within that chemical structure. Well, when we take that helical shape, we can see it inside this three-dimensional structure that we're looking at. So it is still there. It's just we're not so focused on only on that 3D shape. Any questions so far? I don't know if it was my mic, someone else's mic that was picking up. Any questions about this? So now we get to the what is going to be the extra credit question, and this is a good chance for you to consider how you would tackle, and you don't need to answer to me all of the question, but you can make a start of throwing out your part of the answer and then challenging your peers to complete other parts of the answer. So this way I will not provide you the full answer, but I will guide you towards the right answer. So one of the things that we saw is that when you change the chemical structure of a protein, also the physical shape of a protein changed. And we saw that when we talked about Nickel-Kidman. And the question in the exam is going to be that what does Nickel-Kidman have to do with proteins? So again, you can just unmute yourself, shout out, or you can type a little bit of an answer to the chat, and your answer will count as your attendance for this session. Or you can just simply choose to watch in quiet. It's up to you. I will not expect anyone to write a full answer, just what would be the important elements that would help you to start to tackle. It is a short essay that I'm asking you to write, so I'm looking for, let's say, 3-4 sentences, nothing highly in depth. This way also you have known it ahead of the time, so if you really are eager, you can kind of prepare something on a notepad or Word document or Pages document that you just copy paste to the blackboard when you're taking the exam. But what does Nickel-Kidman have to do with proteins? And I'm just going to jump out of sharing the screen because as I've shared before, I'm not able to do both at the same time, see what you're typing and what's going on. I see really good answers there. I think that everyone who has posted has a good understanding of where we're going with that. So let me just share how I would tackle answering it, but that's not the only way. I see probably better answers than what I would provide out there. But if you did not post anything, this would be a good time to at least post your name so you do get attendance for today's session. So how I would tackle this is by reminding the reader that, of course, hair was made of protein. So that's a good starting point for us to start to understand what's going on here. The other thing that we end up noticing is that protein had this primary, secondary, and tertiary structure. Thinking of the primary, secondary, and tertiary structure led us to understand that when we change the chemical composition of the protein, even just slightest of bit, we ended up changing the physical appearance of the protein as well. So while Nicole Kidman went and got a perm, that changed, which is basically altering the chemistry of the hair. That changed also the appearance of the hair. So really, that's all there is. By chemical alteration of a protein structure, we are changing the physical appearance of the hair. I know that it's a little bit of a weird question. It's a great question for seeing if you've been in a class or not, because you will not work it out yourself otherwise. I dare that I've used this question for a while, and I still cannot find online an answer. If you start Googling it, you kind of do need to at least attend to the class to get that. So extra credit question is not something I'm requiring you to answer. It's simply there for you to get extra points. Usually I go about 1 letter grades worth of, but that can vary a little bit. So depending on the quality of the answers as well. So that's what you'll be faced with as the extra credit question for this week's exam. You will find a study guide for the exam in the learning modules between week 6 and week 8, as we discussed at the very start. It is going to be this blank version of the study guide, and we'll go through the answers to the blanks today. Other thing that you'll find, of course, is going to be the actual lecture exam. And I'm really encouraging you to try to take it on time. You have one time to take it. You can't start it, save it, come back later. Once you start it, you have 2 hours to complete it. I realize that sometimes there are IT issues, sometimes internet might go out, and so on. If that happens to you, you just contact me, and I usually should be able to reset it to you real fast. Like I said, 50 questions, more than ample of time to tackle those, and 3 chapters that you're being asked. As a side note, I do have to say that most courses, most other instructors, as you move ahead, I would not be expecting them to provide you study guides. So we're kind of taking an easy start on this course. I want to make sure that you have all the tools for success, but don't get upset with your instructors if they don't provide. That's a little bit going above what we are expecting from the faculty. So other than that, the only last thing that we have left. Perfect, Brittany, you totally got that. She changed her hair structure by changing the chemistry of it. So only thing that we have left is to go through the study guide. So if you did not get a chance to print it out for yourself or open it for yourself, this might be a good time to pull it up. And I'm gonna open it here on the screen and we'll work through it together. This will be rather fast because there is a lot of material to cover, and all of this material should be revision. So I will not have a chance to stop as often as I normally would be to ask any questions. So if you have any questions, just shout out. Help me to spot that. I want to make this time count and be useful for you. So if I'm moving too fast, let me know. Otherwise, I assume that we're going on a good speed. So here we have the study guide and everything that I want to be discussing about it. And we will start with the chapter 3. And the chapter 3 was all about biological macromolecules. And what we end up noticing is that there are 4 macromolecules, 4 types of molecules that are especially important that we end up noticing that are present naturally in nature. First of these were carbohydrates. The second ones that we're going to be talking about are going to be our lipids. And again, I'm just going to try to navigate my way through so I don't lose all of the tools. So we're also going to be talking about lipids. And as the third type of macromolecules that we find naturally, we're going to have our proteins. And I guess that the logical question to ask here would be that, well, does this order matter? Why do I care that you listed it as 1, 2, 3, 4, or 1, 2, 3? The order does indeed matter. The order that we've been shown here is the order in which the body prefers to utilize these macromolecules. So if you have carbohydrates available in your body, those are the immediate source of resources that the body is going to go for. Only if you don't have carbohydrates available, we move to lipids. So this is why losing fat cells, losing fat from your body is so difficult, because if there are carbs, we still keep using those before we go for that extra fat in the body and burning it off. And of course, we remember that if your diet doesn't include enough carbohydrates, lipids, or even proteins, well, then you're going to have to find those resources from somewhere. And what body starts to do, body starts to burn off its own proteins. And the biggest source of proteins that we have in the body, it is, of course, the muscular structural protein. So you might end up getting with muscle wastage, muscle shrinkage, if you do not have any sources for body to do to gain its energy from, get resources from. So then it might be that that's what happens. The fourth type of macromolecules that we really didn't talk that much about, but I wanted to mention, and we will talk about loads of them towards the end of this course, is of course our nucleotides. And where you might have seen nucleotides are when we talk about DNA and RNA. So both of those are really good examples of the importance of these basic macromolecules that we find in the body. Before we move on too much further, there are a couple of terms that I want to make sure that we are comfortable with. And first one of these is the definition of a molecule. So you remember molecule one, atoms that are connected to each other. They were bound together by chemical bonds. And there needs to be at least two atoms to form a molecule. There can be way more than two atoms, but at least two are needed to form the smallest type of molecule that we can have. The other thing that we end up noticing is that usually these molecules that we discuss are going to end up being overall electrically neutral. The other terminological thing that's going to help you as we work through the material on this chapter, if you're clear about it, and that's why I'm taking a little bit of time to discuss about that, is the difference between monomers, in which we only have one single unit of something that we're studying. So here, for example, we would have a single glucose unit, single sugar unit that we're looking at. Polymer instead looks at many of these units bound together. So just so that you are clear between those terms, of course, we can have also dimers, so 2 units, and so on. But when I use those terms, I'm assuming that we're able to grasp that information. Couple of processes that are important as well for us to have appreciation of, and on here I'm going to have to zoom out a little bit so that we can see everything that I want to show on one page. And we're going to start with the process where we take monomers, such as here, individual units. So these are not connected to each other, they're just floating around in the same neighborhood. And we're joining them together to form polymers. So as a result of this reaction, we are left with a polymer. So we have now combined individual monomers into one larger polymer. The other thing that you'll end up noticing is that we are left with byproduct of water as well. So that's an important thing to notice, that whenever we're combining monomers into a polymer, we should have water left as a byproduct of this reaction. And this kind of a reaction is known as synthesis of something, since we are making something larger out of individual components. And in this case, it's going to be dehydration synthesis, since as a byproduct of that, we are left with water. So hydration referring to the water, and D referring that that's the extra element that we're left with. So that was the first type of reaction that we saw. The second type of reaction that we're going to be looking at is going to be almost reverse of that, if you wish to think about it in that manner. So what we have here, to start with, we have a polymer. So we start with a polymer and we break polymer into individual monomers. So we go from one molecule into multiple smaller molecules. And to do this process, we are going to need to add water into this reaction. So water is again important. So let's have a look of the name of the reaction and how the name really explains what happens. So whenever we're breaking something apart, we are talking about lysis. And in this case, because we're using water to break a polymer into monomers, it's going to be our hydrolysis. We're breaking a polymer into monomers with the help of a water. So water needs to be added to that reaction. I hope those make sense. And the next one that I want us to pay a little bit of time is going to be a discussion of enzymes. Why enzymes matter, why are they important for us, and so forth. So let's start by defining what an enzyme is. And this is where you have to pay a little bit of attention. Some textbooks get confused about this, but we don't care about that. We're going to do it the right way. So what enzymes do, they assist in chemical reactions that would happen anyways. So the example of this chemical reaction that I have here is going to be this block, and I'm just going to use a different color for that for the sake of clarity. This block that we see here, that would get broken over time into two separate parts, even if we didn't involved at all. That's just something that would happen anyway. So that would be the chemical reaction that I'm using as an example. So that would happen whether we get involved or whether we do not get involved. But by bringing an enzyme into this reaction, we are now assisting it to happen. And what you noticed with the enzyme was that enzymes had this active site, which meant an area where the material that we're working with can easily bind to. The other thing that we saw is that the enzyme changed its shape while it was doing its job, but then returned back to the original shape at the end of the reaction. So enzymes assist in chemical reactions, and this can be many different types of assistance. It can be helping to speed up the reaction, but it can also be other types of assistance. And once the enzyme has done its job, it can be reused again and again and again. And where a lot of this confusion, I guess, comes from is that textbooks end up using catalyst as an example of an enzyme. Catalyst is a great example of an enzyme, but it's not the only type of assistance that the enzymes can do. So if you do have a catalyst, what catalyst does is it's an example of one type of an enzyme, and what catalysts do, they speed up these reactions. So catalysts are enzymes that speed up reactions. However, not all enzymes are catalysts. Another example of type of assistance that we can have, for example, would be what we discussed here. So here what we are seeing, we're seeing a graph that maps out the energy needed for a reaction to happen in relation to the time. So often in our diagrams, time is on the x-axis, and whatever the other unit that we're looking at is on the y-axis. So often the time just tends to proceed in a horizontal manner on our diagrams. And what we're seeing in this diagram is that initially, normally if we don't get involved, there needs to be a certain amount of energy that gets met before the reaction really takes off and happens. And unless we meet this energy requirement that we would have here, the reaction simply will not take place. Well, but we can help out. We can help out by adding an enzyme into this reaction. And in this case, what the enzyme does, the enzyme brings down this required energy. And even a smaller amount of energy is enough to make the reaction to happen. So really, it's not about speeding it up. We see that the time remains consistent, but we're rather making the reaction happen with a lesser of an energy investment. So that was one example of how an enzyme can help to bring down the activation energy in the reaction. So those were the basic concepts that we needed to know, and now we're ready to go through each of these 4 important biological macromolecules that we need to know about. If you do have questions, just shout out, because I'm not able to see the chat, just because I'm sharing screen. Just even shout out that check the chat, and I will gladly do so. So in terms of the carbohydrates, what do we need to know about the carbohydrates? Well, some good questions would be the sources of carbs, and often students are really good about listing things such as bread, potatoes, pastas, rice, corn, sugars, and so on. But remember that this also includes a large group of various different kinds of vegetables. So a lot of the green stuff can be good sources of carbs as well. One thing that we notice that the carbohydrates are used is for energy for our body to get the energy that can be converted into ATP, and that can then drive the reactions. The other thing that carbohydrates are good, that they also provide raw materials for many different things that we might want to build in the body. So I would know both of those as what carbohydrates are being used for. One more thing that I do want to list when we are talking about carbohydrates is fibers. So fiber is really something that we get from our diet, and even though we get it from our diet, we don't really use it. It goes simply through the digestive tract. So you might wonder, why would it be useful to get fiber in your diet? Well, there's a few different answers. First of all is that fibers help us to give bulk for your digestive tract. So what we end up seeing, and this is a really crappy drawing of a stomach, but bear with me. That's a basic stomach that we see. And what we end up noticing is that on the walls of the stomach, there's actually physical nerve fibers that sense whether the stomach is full, so whether the wall is stretched, or whether it's empty, so the wall is not stretched. And what we can accomplish by eating food that's rich in fiber is to stretch this stomach wall, even if we don't go overboard and eat too much. So we're getting this physical sensation of being full, even if we haven't eaten to the point that our stomach is about to rip because it's too full. So that's the first rule. The other thing that fiber does, it gives us a way for our digestive tract to have something to push against. So it helps the material to move along the tract. And by doing this, helping to push the material along the tract, it helps us also clean or scrape the walls of the digestive tract. So it is important to get this bulk from the diet. So those are the things that I do want you to know about the fiber, and why fiber would be an important part of the diet. We were already looking a little bit about that not all carbs are the same. Carbs can be varied based on the number of subunits that we have here. So when we're talking about carbs, we're talking about saccharides, and monosaccharides were made of one single unit, whereas disaccharides, well, not surprisingly, 2 units, and polysaccharides made of more than 2 units, and this chain could be really, 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 really long. So does it matter which types of saccharides do you get in your diet? I would say that that depends a little bit who you ask from, but as a rule of thumb, I would say that we prefer to eat more complex carbohydrates, so these polysaccharides. The reason is that if we get only mono or disaccharides from our diet, your body goes crazy and burns those off immediately. So then if you need later on more carbohydrates, they're no longer stored there. We've actually already burnt them off. So by eating these more complex carbs, we ensure that they are going to be sticking around for whenever they're needed. And one more term that I want to highlight here is our chitin. So these are one type of polysaccharides, and these particular polysaccharides are exoskeletons of many insects are made of. And later on this course, we will do a lab where we will talk about cockroaches and their exoskeleton and so on. So I will assume that at that time you know what chitin is, that's a type of polysaccharide, and that's what their exoskeletons are made of. So that's the reason why I want you to know it at this point. Nothing deeper than that, but something that kind of might help you later on knowing. And best I can do in terms of drawing it at the moment is that this is our cockroach that we will see on later on the chapters. I already kind of mentioned this part about good and bad carbohydrates, if you wish. And what we saw is that we had a preference definitely for the more complex carbohydrates. Example of this would be whole wheat bread, vegetables, porridge. So it takes more steps to go through these chains of multiple subunits of saccharides. So because it takes more steps, this process will take longer time, and that will ensure that we have these available for whenever you might actually need these resources. If you have just simple carbohydrates, these are so easily available for your body that they get used almost immediately. So what you tend to get when you eat candies, sodas, donuts, things like that, you get this spike of energy and soon after you crash, and you might even be worse off after that than you were before, because we've really gotten quickly that energy burst and we've used it and now it's gone. So that's what I had to say about the simple versus complex carbohydrates. And the next topic that I wanted to talk about is going to be our lipids. So let's have a look of lipids. And lipids, of course, refer to many different kinds of fats that we might have. So let's talk a little bit about those. And some of the sources of fats, of course, include things that you are probably not a surprise to you: oils, butter, cheese, lard. But there are healthier sources of lipids. For example, very Mediterranean diet, olive oil, nuts, seeds, avocados, fish oils, and so on. So there are differences in fats in terms of what we're eating. If you are asked to describe what lipids are used for, really the biggest thing is there that we're using them to storing energy, and especially in a long-term storage of energy. Lipids, fats in general, are not soluble in water, so they are insoluble. And this can become very handy. For example, many animals use lipids on their coats to form this barrier between water and them, or tree leaves can have this shiny lipid coat on surface of them to protect them physically, also from radiation and also from loss of water a little bit. When we talk about lipids, we do need to remind ourselves that lipids are made of triglycerides. So let's have a look of that part of the description. So the glyceride part is what our three fat, or glycerol part is what our three fatty acid chains are attached to. So I've circled the glycerol with kind of a pinkish color, and then you of course remember that we have three fatty acid chains that we have in each triglyceride molecule. What we can have is that not all fatty acids are the same, so I want to discuss that just briefly as a reminder. The first types that we have that we're going to look at are going to be our saturated fatty acids. In saturated fatty acids, what we have is that we have single bonds between carbon atoms. So these carbons are connected to each other with just single bonds. Why this is important for us and why I want to draw attention to this is that this type of saturated fatty acids are typically of an animal source. They are solid in room temperature, and they come with an increased risk of cardiovascular diseases. So at large, we would say that they are less ideal. Unsaturated fatty acids, instead, what we see on them is that there's either one or several double bonds somewhere along the path of the fatty acid. And by having this double bond, there is a significant effect that comes out of it. Well, this double bond causes the structure of this fatty acid to have this kink in the shape of them, whereas the saturated fatty acids were straight chains, whenever there's a double bond that causes that chain to have a little bit of a bend into it. These are typically, and I'm trying to get my tools out here. These are typically liquid in room temperature. They are usually of a plant origin as a fats, and are considered as healthier option to saturated fats that we just discussed about. One interesting subgroup that I've mentioned in the class was, of course, our trans fats or hydrogenated fats. These were man-made to be to have liquid vegetable oils, and then we are changing their behavior to make them solid in a room temperature, something that they normally wouldn't do. So we need to apply a little bit of chemistry there, and we thought we were doing good, but turns out that that might not be that great for our health in long term. I also want us to acknowledge the structure of phospholipid. You might have been at the time when we started discussing these thinking that why do I care? Why does this matter? Well, hopefully by this stage, when we've already talked a little bit about cell membranes, you'll see that these were important to know the behavior of different parts of the phospholipids. So what we see is that phospholipids have the head part, and these are known as hydrophilic. So they are attracted to water. So they prefer to be mixed with water or be close to water. Tail parts instead, and here you'll see both saturated and unsaturated fatty acid chain. We end up noticing that these tails are what we call hydrophobic. So they do not like water, they avoid water. And this is what caused the spontaneous arrangements of these phospholipids, that they ended up, just if you have a bunch of them in a liquid, they tend to arrange in a way that the heads are facing towards water and tails are facing towards each other, so that everyone gets to be in conditions where they prefer to be. And that's how we ended up having these cell membranes spontaneously arranging. Next, I do want to talk also really quickly about steroids. Steroids are characterized by four-ringed chemical structure, and they are the basis for hormones. So our body makes hormones, and one of the ingredients that we needed, one of the materials, is going to be cholesterol. Your body does produce all the cholesterol that it needs. You do not need to get extra cholesterol from your diet, but these are what we use to make the hormones. Next, the third type of structure that we want to discuss about is going to be our proteins. So let's have a look of the proteins and some of the sources of proteins. We'll see that it's not going to be something that you wouldn't expect. Of course, meat is a great source of proteins, milk as well, things like eggs, especially egg whites, tofu, and so on and so on. We use proteins for many, many, many purposes. So there's a lot of uses for proteins. Probably the most important use, if we can say that something's more important than another one, is the structural protein. So structural protein is what we use to build muscles, for example. A thing to remember is that proteins that we get from diet are not really stored for a longer time in the body, just floating around as a free protein. It goes through. So if you are actively consuming protein and you know you will be consuming protein, it might be good to have that. Hello, kitty. Might be good to have a lot of that available from your diet. So you'll end up noticing that people who are really into bodybuilding and things like that, they end up consuming protein supplements while they're training. What protein is then? That I think would make a good question. And this brings us a little bit away from this chapter that we've been having a look of and to the direction of the later chapters that we've already had a look. You remembered a lot of the time when I drew a cell membrane, I also drew proteins on those. So proteins serve many purposes. But what are proteins made of? They are simply chains of amino acids. So again, remember that we can study protein on many levels. The primary level, the structure level would be just as a chain of amino acids. But as we move to other levels of investigation of proteins, we end up seeing that these proteins actually take a three-dimensional shape. What else could I say? This is where our extra credit question steps in. And since we have already taken some time today to talk about it, I'm not going to go through it again. If you're confused, refer back to the video of that chapter. The other example that I do want to mention is that a good example of proteins is our red blood cells. So red blood cells are typically these flattened disc-shaped structures. So it's almost like taking a balloon and then squeezing it from the middle. So it's a little thinner from the middle, but it's still, regardless, kind of this rounded structure in shape. This is what happens normally with the normal red blood cells. We can have also sickled red blood cells where we have an error of a single base in the DNA that codes for the red blood cells, and that single base causes one amino acid to be different from what it should be, and the entire shape of the red blood cell changes. Now it becomes this crescent moon shaped, and that will have significant consequences on the ability of this red blood cell to carry oxygen being diminished. These red blood cells tend to get caught into places where red blood cells would normally flow nicely through, and so on and so on. I'm gonna apologize and just see, you okay? You okay, buddy? Just take it easy. It's okay. So that's what I was going to say about the proteins. And the last one we have left, and I'm simply going to mention at this time, we're not diving into this into deeper discussion, is our nucleic acids. And of course, I think I shared with you that I'm not a native English speaker, as you can work out. So I never really paid attention when I talked about DNA and RNA. But of course, the Na refers to nucleic acid. So we have different kinds of nucleic acids, either for storing genetic information or then translating that genetic information into something useful. And this useful thing will be, of course, proteins. Are you going to find a place to hang out, or are you just going to wander around? So that's our first chapter, and everything that I think you need to know about those. I did have on that chapter, I also went through a nucleotide, so single element that makes these nucleic acids true with you as well. And what we end up seeing when we look through the structure of a single nucleotide is that we see again this familiar shape of a sugar that we've seen before. And within that sugar, we have attached as well a phosphate group. And you will always find the sugar and phosphate in every type of nucleotide. It doesn't matter what nucleotide it is, it always is going to have a sugar and phosphate. So actually, when we are drawing these complex diagrams, what we end up noticing is that the sugar and phosphate form the backbone of the DNA and RNA. It's only the bases that end up varying from one nucleotide to another, but this basic structure is consistent. So really, even though it's consistent and it's an important part of it, the biggest variation and biggest effect that the variation has is, of course, due to the varying bases. And we have 4 different kinds of bases that we can find in the DNA. These can be either adenine, guanine, cytosine, or thymine, so A, G, C, or T. And we'll talk about how these are complementary pairs, so one always match with only one other kind. So they always have to be these matches. RNA is very, very similar, with the exception rather than having thymine as a base, RNA now has uracil. So instead of T, we have U. And again, we will talk much more about these later on this course. At this point, simply the big thing to notice is the DNA is, of course, this double helix structure. So 2 strands that are kind of twisted around each other, almost like you would take a step ladder and rotate that, whereas RNA then was just a single strand. And DNA had within the nucleus, whereas RNA could leave then the nucleus so that the coding for proteins can take place in the cytoplasm. That brings us to talk about our chapter 4, which is all about the study of the cells. And I'm going to go through this a little faster. I think that this is something that you might remember a little better. It's not been that far since we talked about it. So let's have a look of why we started with the cells. Well, cells were, of course, the basic unit of life, the smallest level of organisms that are capable of sustaining their own life, so existing itself. So if we had anything smaller than a cell, it needed to have a bunch of other things to exist. But cell now can exist on its own. So a cell is really the simplest form of life that we can see. And of course, you remember life was all about what we were on this course. We can study cells with microscopy. We had a light microscopy and electron microscopy. Light microscopy is what you get to do. Electron microscopy requires a little bit more expensive machinery that we don't typically have available for you. It is useful, though, to have some sort of an idea about the scale of life, a scale of things that we can see. So human eye typically is capable of telling apart. Hair is about the limit of it. We can sometimes be able to see a cell with naked eye, but that's about it. We can't see much more. So when we recruit a way of expanding the beams of light that we see. So vision is nothing but beams of light coming to your eye. But if we use lenses in a clever way, we can now expand what we're seeing. We're seeing the same stuff that was small here. We're seeing it in a bigger scale. And by doing so, we're now able to take more detail apart of that. So we basically just bend light to be able to see much more. So we can definitely see cells with light microscopes, and we can see many types of bacteria also with light microscopes. However, you will not be seeing the cell organelles or smallest molecules, or let alone you would not be seeing atoms with light microscopes. To do that, you will need an electron microscope. And rather than using the light to study the object, electron microscopes shoot electrons to the sample that we're studying. So that's why it gets a little bit more advanced as a technique, and that's why we don't have it available as well. I'm interested of the limits of electron microscopy, and we are able to take pictures of single atoms nowadays. There's a lot of work going on imaging individual subatomic particles, and that's something that we're very, very interested, but not something that I would expect you to know about. When we talk about the cells and the size of the cells, one thing that we end up noticing is that cells are always small. And the key reason for that was this concept of surface area to volume ratio. So as the cell size grows, the volume of that same grows much quicker than the surface area of that cell. So cell needs to have a certain amount of surface area for materials to come in and leave. If we make a cell really large, it doesn't have enough surface area to serve all of that volume inside it. We can do some adaptations, such as make various different kinds of folds on the cell membrane. That might help a little bit, but even that can do only so much. So for that reason, we need to keep the cells small in size. So otherwise, the metabolic functions, reactions that take place on the cell membrane, there's just not enough cell membrane to serve that volume. Also, the transportation distances within the cell, now, if the cell is huge, grow too large. It might take too long of a time to get from the cell membrane to the inner parts of the cell. So that could be an issue as well. Like I mentioned already earlier on, there are two ways to increase the surface area of the cell. These membrane folds are a really good way of doing that. And of course, it doesn't have to be always just membrane folds. They can be smaller structures known as microvilli as well. So both would be good ways to increase the cell's surface area. Couple of terms that I do want you to know and want you to be familiar is that when we talk about study of cells themselves, we talk about cytology. When we talk about study of tissues, so clump of cells, group of cells, we talk about histology. Are you going to come up? Will that calm you down? So two terms that are worth of knowing, especially if someone introduces their profession to you, that they are, for example, a histologist. You want to know whether they're interested in cells or tissues. One thing that we discussed is that where did the first cells come from? And we don't have a definite answer, but our best theory at the moment is that we had a symbiosis of something that sneaked inside the cell, and both this organelle and the cell figured out that this is very convenient, this is very useful, let's stick together, and more advanced cells started to develop. The next thing that I do want to talk just briefly is the difference between two types of cells that we can have. And we're going to start with something known as prokaryotes. Prokaryotes are often, when I'm describing them to students, they are described as being the simpler. And by being simpler, what I mean is that there really is a cell wall, but there is no nucleus. The DNA just floats around free within the cytoplasm. You can probably see some ribosomes floating there as well, but that's about it. They're very, very simple cells. They are also small in size, so there's not a huge amount of complexity there. I'm just trying to change the highlighter color, but that's not really working out. They were first to evolve, they were simpler, they're smaller. The DNA, the big thing that we didn't have a nucleus. That's something to notice. Let's compare them to more advanced cell type that we can have. And this more advanced cell type that I want to discuss about is going to be our eukaryotic cells. These eukaryotic cells are characterized by a bunch of things. For example, we just see that they're physically much larger than the prokaryotes. They also have a lot of adaptations. They have a membrane enclosed nucleus. They have many cell organelles within there. These different organelles, what we saw when we went through those is that many of those had membranes around them. So they are membrane enclosed structure. These membranes become very important there. These evolved much later in life, so it took a lot longer for these to evolve. Like I mentioned, they're larger in physical size, they are much more complex. We do now find a separate structure of a nucleus, so DNA is enclosed inside the nucleus, and there's a bunch of different compartments that form the cell organelles that we see. And we also took this chapter as a chance to go through the different cell parts. And the first part that we're going to start with by discussing about is going to be our plasma membrane. And you remember that whenever we talked about plasma membrane or phospholipid bilayer or cell membrane, it all meant the same thing. So there's many names for the same stuff when the stuff is very important. But the big thing to remember, if we describe it as a phospholipid bilayer, 2 layers of phospholipids that are packed facing against of each other. There could be other stuff there as well, but that's where the name of phospholipid bilayer comes from. The other thing that we saw sometimes, we can describe plasma membranes as having a fluid mosaic structure. Well, the fluidity refers to the fact that these are constantly changing, constantly materials moving, their organization, their structure is constantly being varied. So they're far cry from those stationary images that you see in your textbooks and on your lecture notes. They are very, very vivid structures. The term mosaic referred to the fact that we had different components that made up the plasma membrane. So rather than just what we are seeing here, this phospholipid bilayer, remember we had proteins embedded in there, we had carbohydrates, we might have cholesterol, and so on. So there's a lot more in there than just this double membrane of phospholipids. We do need to, though, talk about the phospholipid a little bit to highlight the different parts of the phospholipid. So let's start by reminding that we had our phosphate group. So our phosphate group here, that was attached to a glycerol. And from there, of course, we had also 2 fatty acid chains that these 3 parts made up our phospholipid. And I do realize that at this point, some of you might need to jump out. If you do, you will see rest of the session as a recording. So I do understand if you have to go, but I do want to take anyone who's able to stick around, take the entire chapter material and go through it. So don't feel bad if you have to go. The other thing that we saw with the phospholipids is that they had a head part and they had the tail part. And that was important because of how the head and tail behaved so differently. So we remember that the head part was hydrophilic, so attracted to the water, whereas the tail parts were hydrophobic, so they avoided the water. So two important terms to remember there. In terms of the phospholipid bilayer, I think we've already talked about this, so I'm not gonna dwell on it anymore. So two layers of phospholipids, 1, 2, and we had the tails facing inwards and heads facing outwards. Tails away from water and heads towards the water. One thing that we can sometimes see in the phospholipid bilayers, and again, we've already talked a little bit about that, that we had many different kinds of folds or microvilli. The goal of that was really just to increase the surface area of the cell membrane. The next one that we should talk about is going to be our proteins, and I think that we've already touched upon nicely on proteins, so I'm just simply gonna add them to this diagram that we have here. For example, what we saw, some of the proteins went through the entire distance of the cell membrane, whereas others did not and were only on one or another side, whether they're integral or peripheral proteins was the concept there. Proteins serve many different functions, so it really depends on what we're looking. Typically, they can be channels, so kind of tunnels through the cell membrane. They can help us to transport materials. They can help with recognizing other cells, other parts of your body. They can function as receptors for hormones, for example, chemical messengers. They can function like enzymes, or they can be ways to connect cells to each other. What we remember also is that cell has to be filled with something, and this stuff that the cells are filled with is of course going to be our cytoplasm. So this is all the material within the cell other than the nucleus. And it really is this clearish jelly-like stuff that fills the cell. So again, if we sketch just real fast a diagram of a cell here, and we have our nucleus, cell wall, some structures there, and so on. Everything but the nucleus there is going to be our within our cytoplasm. So cytoplasm forms the bulk of the cell, and that's where everything else is found at. It really fills the cell, gives it shape, gives it structure, helps to keep the organelles where they should be. The next structure that I do want to talk about, which is really important structure, is going to be our nucleus. So nucleus is where the genetic information is being stored at. So your DNA has to stay within the nucleus. So it is enclosed into this membrane, double membrane. Known as nuclear envelope. There are small holes through which material can exit and enter the nucleus. These are known as nuclear pores. So if we were to look at the nucleus and the membrane of itself, you'll notice that there are little dots. And I think that on our lecture material, I had some rather good microscopy images of the nuclear envelope, so you could actually really see those tiny holes in there. Well, we remember that the DNA, that's our genes that we wanted to talk when we talked about that. Genes are simply section of a DNA, section that gives us instructions for making a particular protein. So we can't really just come up with proteins and start doing them, we need to have a recipe for making those. And this recipe, if you wish, is what those proteins were. They were instructions for making a protein. Chromatin referred to the mixture of protein, DNA, and RNA. So it's what we will see condensed when we are studying cells at different times of its life cycle later on this chapter or on this course as well. Towards the end of our course, we really focus on the genetic foundation of life. I did talk quickly about how we go from DNA into a protein. So let's just remind ourselves quickly of the basics of that. So DNA was never capable of leaving the nucleus. It has to stay within the nucleus. So inside the nucleus. To get the instructions for making a protein and get those out of the nucleus, what we needed to do, we needed to make a copy of that DNA, and that copy was known as our RNA. And it is this copy that's able to travel in and out of the nucleus of the cell. So we end up finding DNA only within the nucleus, but RNA both within the nucleus and the cytoplasm. When RNA comes together with the ribosome, what we end up getting is a machinery that's able to read that RNA strand and translate the information from that RNA into something useful. And the useful thing that we are translating it in is that 3 bases of the RNA code for a particular amino acid. So it's triplets that we go and get from moving all around the cell, and we put them into right order based on the order told within the RNA chain. And of course, once we had a bunch of amino acids, one after another, that's what gave rise to a protein to take its shape. So this 3-dimensional structure, rather than just the primary structure, which was the just the chain of proteins. So that's how we are able to use the DNA to make a copy of it into an RNA. This process is known as transcription. So think of it this way: when you're transcribing something, you're taking instructions and you're copying them for yourself. So you're really making a transcript of the original document. That's what happens within the nucleus. So the transcription refers to this process where we take DNA and make an RNA copy of it. Translation refers instead to giving kind of meaning to what we have just transcribed. So we're really translating it into something meaningful. This is when we take the RNA and make a protein out of it, or make an amino acid chain out of it. This always has to happen outside the nucleus. Not always. You could have translation also taking place within the nucleus, but that would be less common. So transcription always within the nucleus, translation more often within the cytoplasm. DNA always found only within the nucleus, RNA both within the nucleus and cytoplasm. And it was this process of translation that was taking the instructions that we got and giving them actually some actual meaning to make a protein that we can use. The next structure that we discussed about was our mitochondria. So mitochondria, often for the longest of time, I remember teaching and having been thought of it as the powerhouse or the place where most of the energy of the cell is made. It turns out that ATP is made actually also elsewhere within the cell, but still most of it happens within the mitochondria. But that's what I love about science. We think we know a lot, but still every day, even your basic textbooks are changing. So what mitochondria does, it takes our carbohydrates, it takes oxygen and turns that into ATP. ATP is the chemical formula of energy that can then be used in your body, in your cell, to drive many cell processes. As a byproduct of that, we're producing carbon dioxide. So your cells themselves need to breathe. We need to bring oxygen to the cells. We need to take away carbon dioxide. So this is really the big reason why we're breathing. When we looked at the structure of a mitochondria, you might remember that it was this double membrane sac, and we had these folds inside there. These folds were what we knew as cristae of the of our mitochondria. Interesting thing for the mitochondria is that it does contain its own DNA and also its own ribosomes. So this has some consequences when we do DNA analysis, for example, for forensic purposes. But really the big thing, as we saw, take carbohydrates, take oxygen, turn it into ATP, which was the energy currency, a chemical energy currency. You're left with carbon dioxide and also some water as a byproduct. So that's what the mitochondria does. We'll talk about mitochondria a little bit more in a greater detail later on this course. The other structure that I want to introduce here is a structure, well, we found mitochondria both in plant and animal cells. We end up finding chloroplasts only within the plant cells. And what chloroplasts are, they are made of these double membrane structures, again, two membranes that are filled with this aqueous fluid known as stroma, and there's these disc-like sacs that are piled on top of each other, so as granums. And it is here where we're able to convert solar energy into sugars. So what we do with the chloroplast, we take solar energy, we take carbon dioxide, and we take water, and that can be made into sugars, so into carbohydrates, and as a byproduct we're left with oxygen. So if you look at these formulas that I just wrote for us, comparing what happens in the mitochondria and in the chloroplast, what we end up seeing is that this process is almost identical, only a reverse of each other. So we're making sugars out of solar energy, and we're breaking those sugars into ATP, which was the cellular energy, the energy in a shape that cells can use it. So two almost converse reactions that take place. Another important structure within the cell that we see, known as endoplasmic reticulum, often abbreviated simply as ER. This is a network of various different kinds of tubes within the cytoplasm. Typically, it is continuous with the nucleus. So if we have here our nucleus of the cell with its nuclear membrane, the nuclear envelope, from there we end up often seeing that the endoplasmic reticulum continues from. And you remember that there were two types of endoplasmic reticulum. If there was no ribosomes attached to it, so it's smooth, we call it smooth ER. So there are no ribosomes there. And this smooth ER does many functions. However, if we, yes.
What was F? I don't know if it's your computer or our computer, but it freezes. And then it doesn't
freezes.
scroll down. Yeah.
Okay, our others have the same problem.
Is anybody else having issues?
I am as well.
Anyone?
Yeah, me too.
Okay.
Yeah.
I apologize. I keep looking at my signal strength. How far did you see? How far have you seen? What are you missing?
From F.
From F. So F was chloroplast. Let me try to share it again. I really appreciate that you shouted out, because otherwise I would have just continued, and it's no use for anyone if I just continue and you can't see. Let's try. If I reshare, are you able to see the screen better now? Hold on, I'll stop my video as well in case that helps. Are you able to see F now?
There you go. Yeah.
Perfect. Okay. You don't need to see me. We are being told that it's really important for students to see us, but really, quite honestly, you know who I am. You don't need to see me. So let's hope that it works better. But if at any stage it freezes, just shout out. I really do appreciate you shouting out. So F was chloroplast. That was the only found within the plants, also in algae. So anything that can do photosynthesis has chloroplasts.
Can you go back?
And from there, we got into the endoplasmic reticulum. And I talked how endoplasmic reticulum, we had smooth endoplasmic reticulum, which was just a membrane itself. And then we could have also rough endoplasmic reticulum. When we talk about rough endoplasmic reticulum, there are ribosomes attached to that part of the ER. And you remember the function of the ribosomes was to make proteins. So on these sites where we have rough ER, these are typically the sites where proteins are being made at. Perfect. Let me just check real quickly. Need E as well. If anyone has that, please let me share the E. Okay, so E is mitochondria. Remember, mitochondria was this kind of a bean-shaped structure where we had double membrane, and then inside it we had these cristae, the folds inside. This was where ATP was made out of carbohydrates. Your mitochondria will also need oxygen, but it takes the carbohydrates and oxygen and makes energy out of that, and carbon dioxide. So that was our mitochondria. And I hope that we're able to get everything, but as you did, please shout out if you're missing something. I really appreciate the peer support, and I really appreciate you shouting out when something went missing. The next structure that I want to talk real fast is going to be our Golgi apparatus. I described Golgi apparatus almost like as a distribution center of a cell. So it is where we receive many types of materials in vesicles. So they're coming to this structure in these vesicles inside these membranes. Vesicles get fused into the Golgi apparatus itself, and within the Golgi apparatus, the material contained by these vesicles gets rearranged, sorted, and repackaged so that new vesicles can leave the Golgi apparatus and go wherever they need to go, wherever they're needed in cells. So really, essentially what it does is that it takes material within the vesicles. This material gets sorted, repackaged, and then sent to other parts of the cells or other parts of the cell. So it really is this processing site for a lot of materials. We have a bunch of many different kinds of vesicles, lysosomes or vacuoles. Vesicles often play a role in transporting materials. So they are a way of transporting materials. Lysosomes typically contain something like digestive enzymes, so help us to break apart whatever lysosome fuses with. And vacuoles are typically found especially in plant cells, and you can think of them as wastelands or dumping grounds where materials get dumped into and stored within when we don't get rid of them in another way from the cell. So what happens sometimes when you have a cell, and I'm going to sketch real quickly a plant cell. Let's imagine that this is a plant cell here. So it's a little bit more structured in its shape. It has a little more firmer walls. We do find our nucleus. We end up finding various other cell structures. But over time, one thing that we will see is that it can get filled with a massive vacuole that contains anything that we can't get rid of from the cell, but we really don't want to keep there. So we generate this almost like a wasteland where we dump all this material into, so the plant cells especially end up with vacuoles that can be almost taking the entire volume of the cell's surface area or cell's volume. So those were a few different kinds of sacs inside the cell. I mentioned these only very briefly, mainly because we're going to be talking about these in great depth later on the course. Centrioles are tubes that play a role when the cell divides into two daughter cells. So when we get to talk about cell division, remember these centrioles. They are really just pair of these cylinder tubes that we end up finding, usually located close to the nucleus. They help to pull the contents of the nucleus into two. And of course, cytoskeleton is something that we should acknowledge as an element of the cell as well. So this kind of a scaffolding network made of many different kinds of small microscopic tubes, filaments that fill the cytoplasm. So they serve twofold purpose. First of all, for this cell that we have here, and again, a nucleus, some other cell organelles there. This cytoskeleton really gives the cell its shape. You can think of it almost like as a skeleton of that cell, or if anyone works in the construction business, at one stage in my life, I ended up working a lot with our conservation of stone walls, especially exteriors of the houses. So the scaffolding that's built around the building in that case, that would be a good comparison to what cytoskeleton is, but inside. So it's all these tubes that keep the cell in its shape. And they also allow for the materials to be moved along them. So rather than just floating within the cytoplasm, material is able to travel within the cell thanks to these cytoskeleton pathways or highways, if you wish, within the cell. And the final thing on this chapter that I had, I discussed about 3 different kinds of cell junctions that you could have. You could have this kind of a zipper appearing connection between 2 cells made of many individual points where the attachments are. would be an example of a tight junction. Desmosomes, or sometimes also discussed also as adhesion junctions, are these anchoring spots that are thickened on both sides, on both cells, and from those we have these filaments. So they're almost like these spots where all of these filaments can connect to. Also, these two connect to each other through this space between the two cells. And final one that I had here were gap junctions. They're like these channels that go through the entire space on both sides of the two cells. So easy way for passing materials from one cell to another. So these were our gap junctions. So that's everything from the chapter 4. And like I said, I know that we've ran a little over. I hope though that if we are running over, that you're able to see everything. So again, if for some reason there's any technical issues, just let me know and we'll try to figure it out. I don't want you to just listen to me. I want you to see what I'm showing. What I'm going to do last, I'm going to discuss the plasma membrane just quickly. We're not going to spend huge amounts of time on that. So let's have a look of the chapter 5. You remember I shared with you that so now we're focusing a little bit more in detail to one of the structures that we discussed, and the structure is of course going to be the plasma membrane. So you remember there were many names that we can use when we discuss these structures. And these names really were indicative of the fact that they serve so many different purposes. They're so important for us. Probably the most important function of the plasma membranes is that they're selectively permeable, so they choose certain materials to be able to enter and certain materials not to be able to enter through them, or exit, or not to be able to exit. They are constantly changing. They serve important role also in us recognizing cells, if there's foreign cells that shouldn't be in your body, and passing messages. We discussed quite quickly about the history of how we came up with our current model of the plasma membrane. The big things to take away from that discussion was that already in early 1900s, we knew that cell membranes were made of lipids and proteins. And our current understanding is actually a rather close to this. So you remember we have our two layers of phospholipids, and then we have some proteins embedded in there. And if you really want to make a comparison to the early railroad track model that we discussed, so it's like the metal parts of the railroad track would be our phospholipids, and then the proteins would be what goes through, so like those wooden beams that hold those two sides together. Proteins really don't hold the plasma membrane together, but these wooden beams certainly would hold the railroad track together. But the biggest thing from that is to notice that you'll see that already early on we had a fairly good understanding of what the cell membrane was made of. The other term, fluid mosaic model of the plasma membranes, we've already discussed about that, how the fluidity refers to the constantly changing, constantly varied nature of the plasma membranes. Materials are constantly moving across, but the structure itself is also constantly changing. And the mosaic term to the fact that it's made of many different components. So there's a lot going on there, and all that comes together. We have discussed already, even on this study guide, about the phospholipid and the different parts that make phospholipids. So if I was revising for a test, since it gets mentioned so many times, that might be something that's important to know. What is a phospholipid made of? Well, we end up seeing that, of course, we have the phosphate head part of a phospholipid. And then we had the glycerol, where the head attached to. And we had two fatty acid chains that formed the tails of the phospholipid, if you wish. And we remember that these could be either saturated or unsaturated. And on saturated fatty acids, we only had single bonds between the carbon atoms, so straight structure, whereas on unsaturated, we had this one double bond that caused this kink in the structure, in the shape of the fatty acid itself. I mentioned the head and tail, so this is going to be more of a recap from that. Remember, head parts were attracted to the water, so hydrophilic, whereas the tails were hydrophobic, avoided the water. When we talk about phospholipid bilayer, remember two layers there. And this phospholipid bilayer was consequence of the spontaneous arrangement of these phospholipids, so that the heads were facing outwards and tails inside. This is what helps us to form not only just the cell membranes themselves, but all these compartments inside the cells as well. So all these cell organelles that we've had a look at. When we talk about proteins, a couple of things to notice there. I've mentioned that we can have integral proteins. These integral proteins extended all the way through the cell membrane, whereas peripheral proteins only on a part of the cell membrane. So not extending all the way through it. Levels of structures of the proteins, I shared that we could study it in many levels. Primary, secondary, and tertiary level, we discussed what the differences between those were. So primary level, just amino acid chain. Secondary level, we had these pleated sheets. And on the tertiary level, we got the true 3D shape of that. Again, I'm not going to spend a single moment more on discussing about the extra credit question. I feel that you either are at this point going to know it or you're not. So if not, go and revise that. We talked about different transport mechanisms through the cell membranes. We're going to start with examples of passive transport. Passive transport referred to the fact that we are not using any energy to drive this transportation. The driving force for this transportation is the concentration gradient. So materials from moving areas of high concentration to areas of low concentration. And first example of that we're going to see is going to be our diffusion. So let's discuss about diffusion a little bit. And here I provided you a diagram that's only partially completed. So if you're taking your notes, you might complete that for the rest. So we see that we have some substances here, and these substances are able to move across the membrane until we end up with the same amount of substances on both sides. So until the concentrations are in balance. The term that we use for that is going to be the equilibrium. So we're moving half of the material across until we get the same amount of material on both sides. So there's no longer high and low side. They are equal on both sides, the substances that have been crossing. that's an example of diffusion happens passively, doesn't require energy. The materials themselves are able to cross the membrane. When we talk about osmosis, rather than it being the materials, now it is the water that moves across the membrane. And this was, of course, because the materials that we're looking, the red dots, they're too large, or for some other reason, unable to pass from one side of the membrane to another side. So what we do, we bring down the amount of water on one side and increase the amount of water on another side until the total volume or total concentration within a certain volume is the same on both sides. So water was the one that moved in osmosis from one side to another. Again, passive type of transportation. We don't invest any energy, and we go from having high and low side to having equal concentration on both sides. So at the end, there is no longer such thing as high and low sides. Again, we are working towards that equilibrium. That osmosis brings us to discuss about different types of tonicities. So this is something that's really important later on when you get to discuss, for example, about blood in the body, blood transfusions, other fluid therapy options that we can have. So that's why I'm introducing it here. So remember, we had our hypotonicity. The term hypotonic, referring, if you wanted to use the mnemonic that I had, the hippo. And again, I know I'm not the best drawer here. Here's our hippo. It might end up looking a little more like a pig, but still hippo, hypo, this big swollen animal. So in hypotonic condition, the fluids, the water is flowing into the cell, flows into the cell. And as a result, the cell grows in size and might eventually even end up bursting. So the cell swells as a result of the hypotonic conditions. The next example that I had was when we had isotonic conditions. Isotonic, you can think of it as the statement that I used, that iso perfect. When you're perfect, you do not need to change. And that's exactly what happens in isotonic cells. There's overall no movement of more water or less water in or out of the cell. It's just the same. And the final example that I had was our hypertonic cells. So again, use those mnemonics if they help you, or come up with your own. If someone's hyper, what do they do? I told you that hyper people that I know, at least, they end up running around, being very active. So they get very skinny as a result of that. There are skinny cells. And in this case, water is flowing out from the cell, and the cell ends up shrinking and shrinking and shrinking, and eventually might even end up drying up. The facilitated transportation, what we're seeing in facilitated transportation, and remember, this is again an example of our passive transport. So transport where we are not investing any energy. These substances that we are studying here, they move across the membrane with the help of a protein. So these proteins help us to move until we no longer have difference between high and low concentrations. The concentration is same on both sides of the membrane. So these proteins are almost like little tunnels that help us to move material across. And again, the goal is going to be to end up with equilibrium, the balanced concentration on both sides. The two examples of active transport that I wanted to discuss with you. Let's start with protein pumps. The big thing to remember here is that we do use some energy. And if we are doing active transport, that is usually referring to the fact that we are moving materials against a gradient. So we're making materials go where they normally wouldn't go. So we have to use energy to move them where they normally wouldn't go. One good example of that is that we end up with a charge difference. So pluses and minuses are no longer equal on both sides. So we're moving all the minuses on one side and all the pluses on one side. So we're getting negative cell charge, for example. And a neuron was a good example of that. What happens in neurons, nerve cells, is that the inside of the nerve cell is slightly negatively charged. And because of this slight negative charge, we're able to now pass electrical impulses from one part of the neuron to another. So it's like an electrical cord thanks to its negative charge inside, just on when it's normal, in its normal shape, on its resting stage. The final example of transportation of material that I discussed was the bulk transport. In bulk transport, we now used vesicles to move materials. This can be because we either need to move a lot of material or the material is too large to pass this cell membrane. And it could be either into the cell, in which case it was endocytosis, or out from the cell, in which case it was exocytosis, out from the cell. So these two types of movements of materials, either in or out of the cell. And that really completes everything that I have on the study guide for you to make a note of, for you to know. Remember, every question in the exam is in one way or another from the study guides. So now that you've gone through it, you should be in a very comfortable space of acing the exam. If you do have any questions, I will be sticking around, but otherwise, I'm just going to wish you very good rest of the evening. I know that we stayed a little longer, so if you stayed until the very end, drop your name to the chat and head off to have a very good rest of the night. I will not be offering a session on Wednesday since we stayed so late today. You can instead watch the remaining of the recording if you had to leave early. That's all we have for tonight. I appreciate you sticking around and I hope you have a good night. If you have questions, I will be around here. Thank you. Have a good night. I appreciate you.
I have a question.
I'll try to answer. Let's have a look. What's on your mind?
I just missed something on the first page.
Okay, let's go there. Let me share my screen and shout out when you see it, or if you do not see it, then shout out in that case as well. Let's see. It seems like my computer is about to freeze, so if I disappear, I will come back, but let's give it a chance and hope that it's able to get its act together. So bear with me as I'm trying to share the screen. Apologies for that. If for some reason I disappear, I promise I will come back in just a matter of minutes. I'm going to the first page, and while we're waiting for it, it should be at least really all set up. Let's see. Come on. I can hear you, but I can't see anything moving on my screen. So I'm just closing down other windows, but if I disappear, I will show up in just a matter of minutes, but I'm giving the computer just a couple of moments to catch up. On the first page, I started by defining molecules as two or more atoms that were bound together by chemical bonds, and typically a molecule is electrically neutral. And we had monomers, which was one unit, and polymers, which were made of many units. In dehydration synthesis, we took monomers and joined them together to form polymer, and as a byproduct, we were left with water. And on hydrolysis, we took a polymer and broke it into monomers, and we needed water to be able to do that. I know that it would be so much easier if you saw it. I'm still hopeful that the computer is gonna come around from this, but in case it doesn't.
I just missed the molecule part. If you could just repeat that, I'm gonna write it down.
The molecule part, the definition of the molecule?
Yes, please.
Yes, so a molecule can be defined as two or more atoms bound together by chemical bonds. Two or more atoms bound together by chemical bonds.
Thank you. And then the enzyme, was it just to assist, or was there more?
Yeah, just assists in chemical reactions. So the definition of the enzyme is that it just assists in chemical reactions. Helps in chemical reactions.
That was
and the example of that was bringing down the activation energy.
Okay, yep, I got that part. Thank you very much.
Awesome. Thank you so much. I'm sorry that the screen never came back, but I hope that I will be able to see you next week. I'll definitely have this figured out by then. And I really appreciate you sticking around.
You're very welcome. Thank you. You have a good night.
Have a good night. Take care. Bye.
Bye-bye.
So I have a question for you, but can it be in private?
Absolutely. Let me just stop the recording. How I'm going to do that, I'm going to disappear for a second and I'm going to show up literally in just 1 minute. So please stick around and we can go through. Perfect. So just stick around. I'll be right back.